So good evening and welcome to the Antimicrobial Resistance edition of CF Live. I'm Lucy Allen, the Director of Research at the Cystic Fibrosis Trust, and tonight I'll be joined by a team of experts who'll be talking to us about what they are doing to help beat antimicrobial resistance in cystic fibrosis. And I'm sure I don't need to explain to those of you that are watching this why antimicrobial resistance is a research priority for us here at the Trust, since the health of people with CF is so dependent on the availability of effective antimicrobial treatments. We know that bugs such as bacteria and fungus can cause nasty infections in CF, but are very clever and they can find ways to adapt in order to defend themselves against antimicrobial drugs, and this is termed antimicrobial resistance, or AMR. I'm now going to hand you over to our team of experts who will talk you through some of the research that they are doing, which has been funded through the CF Trust to help better understand antimicrobial resistance, and how we're working with companies to help try and develop new drugs to fight these tricky infections. And at the end of the presentations, there'll be an opportunity for you to ask questions, so please do use the chat box to ask any questions that you may have. So first up, I'd like to introduce you to Professor Chris van der Gast from Manchester Metropolitan University, where is a Professor of Microbiology. CF has been a focus of Chris's research for about 5 years, and you will be talking to us this evening about how pathogens live alongside other bugs in the CF lungs. Over to you, Chris.
Thanks, Lucy. Okay, so thanks for that. So yeah, I'm Chris van der Gast. I'm a Professor of Microbiology at Manchester Metropolitan, and I'm also an honorary consultant microbiologist within the Northern Care Alliance NHS group in Greater Manchester. And so understanding how pathogens live alongside other bugs in the CF lung is pretty much the connecting theme across all of my CF-related projects within my group, and which many of are supported by the CF Trust. On the next slide, please. And so when we think about infection in the CF lung, we typically think along the lines of a one microbe, one disease kind of a concept of infection pathogenesis, or rather, it's just one pathogen creating the infection. And that's kind of what we can see depicted in this cartoon of Pseudomonas and lots of different cells of Pseudomonas. And so a lot of, I suppose, certainly over the last 20 or so years, a lot of time has been spent looking at pathogens in isolation, like Pseudomonas, to understand the nature of the pathogen, how they react and respond to antimicrobial treatments, and then how they, more worryingly, how they develop antimicrobial resistance through mutation or exchange of genetic material between different cells and then across the Pseudomonas population within the lung. And that's been, that work over the last 20 or so years has kind of really helped to get us where we are now in terms of the treatment that's been able to be offered. But that's not the whole picture. Can we have the next slide, please? The Pseudomonas just doesn't live on its own. It lives in the lung alongside other pathogens, for example, with Burkholderia in the blue there, Staphylococcus in the red, Haemophilus in the pink, and other pathogens which I didn't get around to drawing over the weekend. And all of these are interacting with the cells and with each other and exchanging genetic material, able to allow antimicrobial resistance to kind of spread throughout these species. And if that's not complex enough, can I have the next slide, please? Enter stage left, you've got emerging pathogens like non-tuberculosis Mycobacteria. Now, in terms of these Mycobacteria, we don't really know how they interact with other microorganisms in the lung, and this is kind of the basis of one of my projects that's funded by the CF Trust to kind of start unraveling that process. And if that wasn't complex enough, can I have the next slide, please? We actually have all these microorganisms live in a lung microbiota, a community of many different types of microorganisms, of which pathogens recognized and emerging are embedded in. And they're all interacting with each other in ways that we don't quite understand yet. And in fact, some of these other microorganisms that are present, we're not entirely sure what their roles are. We're trying to work that out. This is the reason for a lot of our work, is to drill down. And so we're trying to work out, are they all pathogens? Are some of them pathogens? Are some of them Yeah, it would be a great fabric design. Are there some of them opportunistic pathogens? Actually, they get an opportunity to become pathogens, but they're not normally. Are some of them just sat there not really doing anything, as in interacting with anyone else? Or so do some of these actually rile up some of the existing pathogens to make them more virulent? We have some evidence to show that actually the presence of some of these unknown types of bacteria, or as unknown in what their functions are, actually mitigate the pathogenic effects of some of these main pathogens. So where we come from in this is, instead of coming from that bottom-up approach of looking at an individual pathogen and working upwards, we're taking a top-down approach to deal with all this complexity and then drill down into that to, I hope, in the end, to provide novel ways of being able to treat these complex infections and further improve the lives of people living with CF. Thank you.
Thank you, Chris, and thank you for a fascinating talk to open our evening. And I'm loving your hand-drawn slides, and absolutely, I'm going to be ordering a pair of curtains from you. Thank you. And so up next, we have William McCarthy, and William's currently in his third and final year of his PhD at the University of Cambridge. William will be talking to us about a really exciting and innovative approach to developing antibiotics for Mycobacterium abscessus, which not only causes really nasty infections in the lungs of people with CF, but is also resistant to many antibiotics. William, over to you.
Thank you, Lucy. I hope everyone can hear me. In the next 5 minutes, I'm going to talk about the research we're doing at the University of Cambridge towards tackling Mycobacterium abscessus infection. Slide, please. M. abscessus is a particularly stubborn bug. It's very good at resisting treatment by antibiotics, and this is due to 4 main defense mechanisms shown here. Firstly, in the top left, the mycobacterial cell wall, which is a shell that surrounds and protects M. abscessus, is particularly difficult for antibiotics to penetrate. Any antibiotic trying to get into M. abscessus has to push through 3 of these distinct barriers. Secondly, M. abscessus is equipped with what we call efflux pumps. These efflux pumps can be likened to water pumps, and they push antibiotics out of the M. abscessus cell, rendering them ineffective. Thirdly, M. abscessus contains enzymes which can be used to break down antibiotics before they can kill the bacterium. Just like a butcher chopping up meat with a cleaver, M. abscessus chops up antibiotics with enzymes. And finally, M. abscessus can actually modify or change the target the antibiotic is going after. It can do this, for example, by modifying the shape of the target so that the antibiotic no longer fits and therefore no longer affects M. abscessus. For these reasons, M. abscessus infection is extremely difficult to treat and routinely requires a year-long course of strong antibiotics to clear. This cocktail of antibiotics can have damaging and unpleasant side effects, and so it is essential that we identify new antibiotics that can treat M. abscessus more effectively. Slide, please. Before talking about the research we're doing at Cambridge, I think it would be useful to give a quick overview of how drug discovery actually happens. You've heard me mention the word target on the previous slide, and this is where the whole drug discovery process really begins. The target carries out a function essential to some cellular process, and thus the survival of the cell or the bacterium. A drug is then designed for that specific target with a view to disrupting its function, thus interrupting the cellular process and subsequently killing the cell. How this is traditionally done is that a pharmaceutical company would have a pool of hundreds of thousands to millions of molecules, which can be tested against the target in a process known as high-throughput screening. High-throughput screening narrows down the number of molecules to the ones most effective against the target, usually leaving up to 1,000 molecules. A number of these molecules are then selected and elaborated into drug candidates following an iterative workflow of designing molecules, making them, and testing them. Potential drug candidates then tested in animal models, and should a drug candidate prove successful at clearing the infection in the animal, it proceeds to clinical trials where it's tested on people infected with M. abscessus. When a molecule passes clinical trials, it's then packaged into a form which can be administered to people as a marketed drug. This whole process can take up to 10 years and cost more than £1 billion. Slide, please. In our research at Cambridge, we're applying a more modern technique to this early stage of drug discovery called fragment-based drug discovery. Fragment-based drug discovery, being an alternative to high-throughput screening, really does simplify the initial stages of drug discovery. The failing of the high-throughput screening approach is that large molecules identified against the target don't actually form the best fit and so aren't the most effective, shown here on the top left. In fragment-based drug discovery, molecules that would normally be tested by high-throughput screening are broken down or fragmented into smaller pieces known as fragments, shown here in green. These fragments are then tested against the target individually, and because they are smaller, they fit more snugly with it. These fragments identified are then stitched together into a molecule that fits the target much better than the molecules identified by high-throughput screening. And this is kind of shown schematically on the left. But furthermore, high-throughput screening tests hundreds of thousands to millions of molecules against the target, whereas in fragment-based drug discovery, a much smaller number of molecules in the range of just 1 to 10,000 can be tested at this first step. As a result, fragment-based drug discovery is much cheaper than high-throughput screening and can be done much quicker, thus speeding up and reducing the cost of drug discovery while also producing higher quality molecules. Slide, please. So getting on to our work, firstly, there are thousands of different chemical reactions taking place in bacteria to help them grow and survive. Existing antibiotics target only a small fraction of these. And as I showed earlier, M. abscessus is quite good at building up resistance to these types of antibiotics. Our work is attempting to target chemical reactions that have never before been addressed. In this way, antibiotics derived from these new targets will have a head start on resistance. We've decided to target coenzyme A production. Coenzyme A is essential to a cell's metabolism and energy production. Our hypothesis is that stopping coenzyme A production will interrupt these two vital processes, resulting in the death of the M. abscessus bug. To do this, we are targeting a specific protein called PPAT, which affects one of the 5 steps by which M. abscessus produces coenzyme A. Over the last 3 years, we've tested a library of 990 fragments against PPAT and successfully identified 19 fragments that fit PPAT well. Through iterative cycles of the design make test workflow, we've managed to stitch together a number of these fragments to produce bigger molecules that have an effect on PPAT. However, these molecules have only a modest effect on killing M. abscessus at the moment. This is a work in progress, and over the remaining year of my PhD, I hope to further improve the best of these to produce molecules that have a significant effect on M. abscessus. Next slide, please. I want to just finish by just kind of reiterating how important it is that we tackle antimicrobial resistance now. In 2015, the UN released a set of Sustainable Development Goals shown on the top of this slide. And these are a collection of 17 interlinked goals designed to be a blueprint to achieve a better and more sustainable future for all. In 2019, the International Coordinating Group on Antimicrobial Resistance identified AMR as a major barrier to the achievement of several of these goals, as I show on the bottom. In these times of pandemic, I don't need to remind anybody how susceptible humanity is to infectious diseases. In 2019, the World Health Organization described antibiotic resistance as an invisible pandemic, and currently at least 700,000 people die each year due to drug-resistant diseases. But the urgency needed in tackling AMR is obvious. Slide, please. Of course, I haven't done all this by myself, and I would like to thank everybody on this slide who has contributed to this research, as well as the CF Trust for funding the work and all of you for listening. Thank you very much.
Thank you, William, and thank you also for the reminder that antimicrobial resistance research is important for global health and not just for research in CF, but obviously we are paving the way within CF, and hopefully findings from our research will have bigger impacts for global health going forwards. So next I'd like to introduce Ronan Murphy. Ronan works at Imperial College London. He's part of Jane David's research lab. And like William, he's also in his final year of his PhD. As part of Ronan's PhD, he's been looking at how to improve the effectiveness of existing antibiotics to Pseudomonas aeruginosa using resistance breaker drugs. So over to you, Ronan.
Very much. Good evening. As Lucy said, I'm a microbiologist and I work in the lab with Pseudomonas aeruginosa. And one of the aims of my research is to try and make antibiotics more effective against Pseudomonas. So slide, please. So my aim is to, using this term, break the resistance of the Pseudomonas to antibiotics. Slide, please. So in chronic infections such as Pseudomonas and CF, we know that antibiotics don't work very well and have some side effects, especially from after long-term use. So we want to know if we can make the same antibiotic doses that are currently used more effective than they are. So can we give something else at the same time as the antibiotic to make it more effective and maybe recover some of its activity against resistant bacteria? Slide, please. So my aim is to, using this term, break the bacteria's resistance, thereby clearing infections with lower, safer concentrations of antibiotic. Next, please. So I work with a drug called glatiramer acetate, we'll call it GA. It's a drug already in use in multiple sclerosis and has been for quite a while. I know from previous work that I've done that it can kill Pseudomonas, although it doesn't do that very well. However, it does look like other known resistance breakers. So, slide please. My question is, will GA break the resistance of Pseudomonas to common antibiotics used in CF? Next slide. So how we look at this initially is we usually start off with looking at how the bacteria grow. So this is an example of normal pseudomonal growth, starting at the bottom left with a low number of bacteria. And then as we move across the graph, we've got increasing bacterial density over time, indicating growth of the bacteria and more bacteria being present. And we get this kind of S-shaped curve that we associate with healthy bacterial growth in liquid media. If we add in a tobramycin dose to this, we see that while there is some effect of the tobramycin in this case, after a while the growth returns to a kind of a normal pattern that you see above and reaches a similar level to the untreated bacteria. Looking at what GA does itself, again, as I mentioned before, we see a small effect of the GA on the bottom left, where the number of bacteria is reduced initially, but after a while this is recovered by the bacteria and it ends up growing normally into a comparable level to the untreated one. However, what we noticed when we combined these two doses of GA and tobramycin together, we got an entirely different growth pattern, leading to a much lower density of bacteria compared to either of the agents alone. So that indicates to us that something is happening here with the two agents that wasn't present when either agent was on its own. So using this observation, we carried forward and we leave these bacteria grow overnight, and then after overnight exposure, we count how many bacteria are left. So for a small dose of tobramycin, you can see the black dot here, we get a pretty high level of bacteria. And when we combine that with glatiramer acetate, we get a decrease in the number of bacteria. While there is a decent drop in the number of bacteria, we still get a high number present after overnight growth. So you see approximately the 95% difference between the tobramycin alone and the tobramycin plus GA, but still a high number of bacteria. If we increase the concentration of tobramycin ever so slightly, but leave the GA at the existing concentration, we see this gap beginning to widen, and now we're killing more than 99% of the bacteria when we have the combination treatment versus tobramycin on its own. And if we increase the concentration again ever so slightly of tobramycin, what we see is that while tobramycin on its own allows the bacteria to grow into the tens of thousands after overnight exposure, the combination of the two things kills all the bacteria. So in the red line here, that's indicating no viable cells after overnight exposure. So in the next slide then, we carried this on to look at this effect in cystic fibrosis Pseudomonas. So I took 12 CF strains of Pseudomonas and measured the lowest concentration of tobramycin that was needed to work against these strains. So that's known as the minimum inhibitory concentration. And then I repeated that test, but added in GA as well at the same time. And what we saw was that GA lowers the concentrations of tobramycin required to have this effect. So less tobramycin was now required to work against the Pseudomonas. So this graph shows each of those 12 strains. So the black dots is each strain with tobramycin on its own, while the red square is the combination of tobramycin plus GA. And you can see from the connecting lines that the majority, although not all of the strains, the concentration is dropping to a lower level than it was when tobramycin was administered alone. And next please, the lowest effective dose was reduced on average by 50% for tobramycin when GA was present rather than when tobramycin was administered alone. Next. So what we've discovered is that we can break the resistance of tobramycin to resistance of Pseudomonas to tobramycin with GA present. We can give a lower concentration of tobramycin, but keep its effectiveness in these lab tests. And we believe that glatiramer acetate is therefore a good candidate for drug repurposing, given that it's already in clinical use in a different condition. So as well as the fantastic support of the CF Trust, this project is also in collaboration and partnership with groups in Aarhus University as well as Cycle Pharma, and we get a lot of great help from the Royal Brompton and Harefield College. And with thanks to my boss, Professor Jane Davies, and all of my colleagues at the CF and Chronic Lung Infection Group at Imperial College London. And thank you for listening.
Thank you, Ronan. It's really exciting to hear how other already licensed drugs are potentially being repurposed to help us tackle AMR, because as Ronan mentioned, that then speeds up the process through which these drugs are developed and hopefully gets them to patients quicker if they're successful and effective. And lastly, taking a slightly different perspective, I'd like to introduce Dr. Deborah Neal. Dr. O'Neill is going to talk to us about an innovative new partnership called the CF Syndicate in Antimicrobial Resistance, which is helping to get some of these new drugs, like the ones William and Ronan are working on, into the clinic more quickly and efficiently. Deborah's chair of the Syndicate Steering Committee. Over to you, Deb.
Thanks very much, Lucy. Hopefully everybody can hear me. So good evening, everyone. As Lucy kindly introduced me, I'm Deborah, and I do currently have the pleasure of chairing the steering committee of the Syndicate. My day job is running a biotechnology company that is developing a range of antibacterial and antifungal therapies for drug-resistant infections, and two of those products actually address infections in cystic fibrosis. It's been fantastic. Before I talk a little bit about the Syndicate, I think that the reason that this exists is supported by the fantastic talks that we've heard tonight, and it's always wonderful to be reminded, I think, not just of the vibrant AMR research community in the UK, but specifically the fantastic research that's ongoing specifically to address infections and drug-resistant infections in CF. And this is fantastic science, but obviously we're very keen to translate science into potential therapies to address infections. And science is really just the beginning of the journey, and there's absolutely no lack of fantastic ideas, great science that could potentially underpin new treatments, but the science is almost the easy bit, and translation of lab data into potential new treatments is a lengthy, it's a complex process involving the regulatory authorities who license medicines, manufacturing, all sorts of different elements to get that science into a product at the end of that journey, hopefully. For antimicrobials, it's particularly complex just because there's no particular set pathway, no two drugs are the same, the target bacteria or fungus for drugs can be very different. They all work in particularly different ways. So it's a minefield, a maze to try and get through and navigate the regulatory process. For drug-resistant infections, it's even more complex because of the bigger ask, if you like, of what the drug has to do. And you've heard some very exciting, very novel approaches discussed this evening. And obviously for CF, you're drilling down even more. CF infections are not like other infections. You have specific pathogens that cause those. The CF airway is unique, so there are lots of other parameters. So there's a lot to think about beyond the science, and really that's why the Syndicate is there. So if I could have the next slide, please. So what we're really trying to do is accelerate that process, that pathway that I was talking about, from the discovery of fantastic science and making sure that that can hopefully be translated and eventually become novel CF antimicrobials in clinical practice, but navigate quickly and effectively through the pathway to get there. And the Syndicate came about from the Medicines Discovery Catapult joining forces with the Trust. And what we do and what the Syndicate is all about is bringing together all stakeholders in this journey, anyone who this is important to, and experts in this area. So we've got industry leaders, We have patients within the working group, as well as the syndicate, clinicians, academics, every bit of expertise that you could imagine. And we have a smaller working group that steers what the syndicate does. It's essential in terms of this coming about from asks. So there are companies, there are research groups out there who were kind of struggling to find out how to start that translation journey. So how to begin to turn their great science into potential therapies. And it became clear that there was a need for the syndicate to hopefully provide some kind of guidance, some kind of resource, which I'll come on to specifically. And as I mentioned, that there's no set path, so the syndicate isn't there to say, well, if you're developing a particular drug, this is exactly, the direction that you should be heading in. But certainly we can point people down a particular path to accelerate that process and make sure that the data that researchers are getting in the early stages before they get into that journey is a fit for what's required later on. And that's essential, because that's certainly going to streamline the research and the translation of potential new therapies into actual drugs at the end of the day. So certainly being able to point to the critical steps that are needed, the things that are potentially common to most pathways, even though no two are the same. And also what I'll talk about is what became clear is some researchers, some companies don't always have access to the cystic fibrosis specific research tools. As I mentioned, the complexity of developing an antimicrobial for CF is even more so than for a non-CF infection, because of the environment in which the drug has to work. CF-specific bacteria, you don't want to develop a CF drug against a bug that doesn't play a role but is just similar to a CF pathogen. So I think access to not just expertise and some guidance, but also some research tools is where the syndicate comes in. So if I could have the final slide, please. So this is just more detail of the areas of focus that we're working on at the moment. And these are the three key things at the moment that we think can address the main challenges and the barriers to researchers developing great science into potential therapies. So first of all, as I mentioned, some of the tools that researchers often don't have, enabling people trying to develop therapies to have access to clinically relevant samples for the early testing of their potential products. So these samples could be CF-specific bacteria, so culture collections of relevant target organisms to test potential drugs against, and those are essential. And believe it or not, sputum, the biological matrix in which these drugs have to work. So these are all incredibly important samples, which often, as we hear from the syndicate, probably isn't explained maybe as much as it should be to the community and the patients who can obviously provide these samples, but they're absolutely essential for anybody that's in this area of research. And as we mentioned as well, back to that pathway and the key experiments that you have to do, making sure that the data that is generated is the best kind of data and that fit for the process and the journey that drugs have to go through, and therefore streamlining really the whole process and reducing the time that it's going to take for that good science to be translated into a drug in the clinic. And then the third thing that we're focusing on at the moment is really the point that you have to work back from when you're thinking about the profile of a new therapy, the specific infection in CF that you want it to address. That obviously involves all stakeholders, clinicians and patients as well. But that helps because that does shape to some degree the research pathway and the early data that researchers will generate as the start of that process to hopefully developing new drugs. So I said on the scoping calls when the Syndicate and the Trust were initially, I guess, doing the feasibility work to see if a syndicate would be worthwhile. And this absolutely holds true with the point that the syndicate's come to now, that, you know, as a go-to, as a resource for anybody working in this field, I think it's wonderful. I think Novobartic's own products had the syndicate that we want to develop been there a good few years ago, probably would have been developed more quickly and in a more streamlined way. So I think it's essential that this is there. And I think it's starting to have an impact from, you know, the number of approaches that the syndicate is getting. I think the research community are beginning to know that we're there now and that we are a resource and a go-to for people to approach. So I will end that there and look forward to taking any questions about the syndicate that folks may have.
Thanks, Deborah, and thank you to all our experts for that amazing whistle-stop tour of all of the research that we're helping you support, and obviously the syndicate. So now it's over to you all to use the chat function to post any questions that you may have. In the meantime, can I perhaps ask all the speakers to turn on their videos if they haven't already done so? And I've got a couple of questions that were submitted during the registration process, and we've also got a couple of questions that have been posted online already as well. So Chris, first question for you. The question is, what are your thoughts on how a better understanding of the lung microbiome in CF can change how antibiotics are prescribed?
Yeah. So that was the point I think I was trying to go across, and I don't know how successful that was. You have multiple bacterial species there that are all able to communicate with each other, but also, you know, you might be targeting kind of one of those microbes with one antibiotic, but it's not effective against the others, or in fact that they're resistant against it. That they develop a resistance that's then transferred across the rest of the organisms therein. So it's trying to, you know, I think it was alluded to in some of the other talks, and Deborah was just putting it across there, is like, how can you target a drug against a given pathogen within that milieu of different bacteria that is effective against that, but doesn't affect everything else, and then knock that whole, create more problems than you had before. And I think that targeted approach is certainly probably one of the more logical ways to go in terms of new antimicrobial agents.
Absolutely, it's a fine balance, isn't it, with antibiotics, between knocking out the good to defeat the bad, etc.
Yeah.
So, yeah,
absolutely.
Okay, next question for Ronan. So this is one that was posted in the chat. The question is, do other antibiotics have the same relationship with GA, or is it just tobramycin?
So far, tobramycin is definitely the strongest effect we've seen, the best sort of co-reaction between the two. There is some indication we have that there may be something happening with ceftazidime as well, which is from a different antibiotic family, although, as I say, not as strong as what we've seen with tobramycin. And I've also tested ciprofloxacin and colistin, neither of which appear to have any extra effect when GA is present.
Okay, thank you. William, question for you. Would the targets that you're developing for M. abscessus also work against other NTM bugs?
Yeah, that's a great question. The short answer is yes. I'll give you a longer one. So we're targeting Coenzyme A production, and Coenzyme A, it's essential to all living organisms. You know, it's essential in us humans. But the thing is, the way that M. abscessus and the way that humans produce Coenzyme A is different enough that we can specifically target the way it's produced in M. abscessus, but not affected in humans. Luckily, the non-tuberculosis Mycobacterium family are kind of closely enough related that should, you know, we develop antimicrobial against Coenzyme A production, it could definitely have an effect on the other non-tuberculosis Mycobacterium. And just kind of to add to what Chris was saying there, it kind of builds into that, targeting specific bacteria over others. If Coenzyme A production was then different, say in Pseudomonas, you might be able to get that specificity that Chris was talking about.
Okay, thank you. And Deborah, a question for you about the syndicate and how people with CF can get involved and help with the research, particularly around providing samples such as sputum samples, etc.
That's a great question, and something actually that I think we've started to discuss more as a working group. So I think I would certainly say at the moment already patients are very much involved in the syndicate and sit on the working group. And one thing that we're trying to do with our clinical experts is work out, I guess, the best pathway to communicate to their patients and the wider community that, potentially when they're providing samples, this is what they could actually be used for. So yeah, I think that communication to the community of how valuable, samples are in research, not just things to be chucked in the bin, is essential. And yeah, that's an ongoing discussion.
Okay, thank you. And just to bring it to everyone's attention, today we launched a research in focus report on antimicrobial resistance, so just in time for today's event. So there's a link in the chat if you want to read more about it. So we've got another question in. I'm not sure who it's for just yet, and the screen has disappeared. So it's a general question. It says, as we think about antimicrobial resistance, are we locked in an eternal arms race, or are there some types of treatment that microbes physically could not evolve resistance against? Anybody want to tackle that one?
Ooh, can I have a first go? Because I'm an immunologist, by the way, I should have said, so I probably stand out like a sore thumb. Yes, I think a lot of what we've heard this evening has been the stuff that's really at the forefront and really trying to address this almost, can we try and overtake bacteria in this arms race and come up with novel approaches that aren't the traditional one antibacterial against one molecular target. And I think looking at these alternative strategies and almost trying to fight infection the way the body would, seems to potentially be a way of getting around that, because obviously we have evolved with bacteria, and the body, and I think certainly the CF lung in particular is phenomenal, just in controlling that amount of microbes. So we've obviously developed strategies to coexist, and I suppose it's harnessing the bits of that control bacteria therapeutically that might be the future. So there are some really exciting, and they're called non-traditional antimicrobial, and you've probably heard this evening that we've not talked about antibacterials too much, but in broader sense is antimicrobials, so that there is some exciting stuff coming. I don't think the arms race will ever end, but hopefully we'll be able to nudge forward and come up with our own tricks to defeat some of these bacteria. Much needed.
Yes, thank you, Deborah. So another question about the process of drug development. So the comment is that we've heard the overall process of drug development is long and expensive. If Ronan and Williams' research is successful, when would we expect to see benefits for patients?
Well, it's still very difficult to predict. We would hope that given GA is already an in-use drug, that it would be sort of slightly closer to the clinic than a drug that's starting from scratch. But it does have sort of a few hurdles to get over yet. So in normal course of things in MS, it's given as an injection subcutaneously, whereas our vision would be that this would be an inhaled treatment in CF. So there's that changing of the formulation of it so that it's now deliverable in that method and that we can get that concentration into the lung. As well as when you change the formula, obviously, while we know and believe it's quite safe, you still have to do the checks to make sure that after changing it, it's still safe and will still be effective. So it's still a few hurdles to get over yet, but we're quite optimistic about it.
I think I can add to that. And talking about our specific strategic research centre, that we're kind of even further behind than Ronan is, because the molecules we're making are trying to come up with new modes of action. And so they would have to pass through clinical trials fresh from the beginning. Another project funded by the SRC has got a compound or a molecule now looking at preclinical studies, but it's still going to take quite a bit of time before that will get to people with CF.
And I guess that's part of the challenge, isn't it? You've got to have enough drugs and potential compounds in the development pipeline, so you've got a constant stream of things feeding through into the clinic. And that's, I guess, where we're trying to focus our attention at the moment, so that we're not just thinking about modulated therapies, it's thinking about the whole package, and where there's still unmet challenges such as AMR. Thank you. Any other questions? I know there is one about phages in the chat, and we will respond to that offline, and we'll send some more information about that. Okay. So thank you again so much. It's been a wonderful evening. Thank you for all of the experts for taking their time to talk to us about their research in this really important area for CF and also for global health. And thank you all for joining us tonight and for asking some really interesting and challenging questions. And please do join us again for our next CF Live event, which will focus on clinical trials, which is the next step on from the research pathway, as we've just heard from Ronan and William. The clinical trials event is being held on the 4th of November, and we'll talk you through what clinical trials are, why they happen, and the different ways you can get involved with them. And we look forward to seeing you then. Thank you.